It's been a wonderfully exciting day, and this is going to continue now. Mark Wightman from the University of North Carolina at Chapel Hill is yet another great technique for studying the brain, and he will tell us about monitoring of neurotransmitters and their metabolites. So let's welcome Mark to the podium.
Thank you very much, Nick, and thank you to all of the audience who have been here all day. It's been a really exciting day hearing all these new techniques. The technique I'm going to tell you about is perhaps is much older than most of the other techniques that have been talked about. I would say the first review on this technique first came out in 1976, believe it or not. But things have developed a lot over the years, and so we have a technique now that can really monitor neurotransmitters, metabolites during behavior, and that's kind of the goal of a lot of our neuroscience sensors. But in addition, now we can start combining this technique with other techniques so we can get a more complete picture of what is actually going on in the brain. So the way we go about monitoring neurotransmitters is by the technique of electrochemistry, a classical technique that you all probably studied in freshman chemistry. The guy that I postdoc for, Ralph Adams, was clever enough to realize that many neurotransmitters, for example, dopamine here, which I'll be using in a lot of my examples here, is not only a neurotransmitter, but it's a molecule that can be oxidized very, very easily to form this orthoquinone. And when you do this oxidation, if you do it in an electrode surface, you end up with electrons. These electrons can be measured as a flow of current, and the amount of current is proportional to the concentration. So that's the approach that we've used here to look at neurotransmitters. You have to be careful in this game. For example, this molecule, dihydroxyphenyl acetic acid, is the major metabolite of dopamine. So everywhere you've got dopamine, you're going to have DOPAC present. And this molecule, its oxidation mechanism is quite similar. But it turns out that we can, in fact, get unique signals from dopamine compared to DOPAC and compared to other substances by using some techniques that I will show you here today. Serotonin, another molecule that's been mentioned here today, is also an electroactive neurotransmitter. It's a substance then that we can use the techniques that I'll tell you about today to actually look at and monitor as things are going along. So the kinds of questions we want to get at are multiple, and I just thought I'd use this little cartoon to illustrate some of them and some of the complexities. So what we're interested in is how dopamine does its job of neuromodulation, and what you're looking at here is a dopamine nerve terminal. It comes near these neurons that are called medium spiny neurons in the ventral striatum and also the dorsal striatum, and it secretes out dopamine, which can interact with receptors over here. And the activation of the receptors doesn't do anything that you can easily measure. It does activate various second messenger pathways. But what it really does is modulate the electrical activity of the cell, so when the main input, the glutamatergic input, comes in, it can modulate what the glutamate actually does. So we want to be able to see dopamine release and understand what caused its release, of course, and be sure that we know that we're looking at dopamine and not some other molecule. But we also want to understand these receptor interactions, and we want to understand the firing rate changes that occur in the neurons, because that's the way information is processed in the brain. The slide that I'm showing you comes actually from Jeff Wicken's lab, and he was trying to illustrate the complexity of this task of looking at neurotransmitters. Because we don't, when we're dealing in the intact behaving animal, have one synapse or synaptic region in isolation. Rather, we're dealing with a number of different substances in this cube that's 50 microns on a side. These different chemical actors are portrayed here by different colors. So there are multiple different substances that are being secreted, that are being used by the neural processes. They're not just neurons, they're glia, they're blood vessels, and so on. And we want to try to understand how this complex matrix is actually working and leading to that endpoint, the behavior of the animal. So, this whole set of problems that I've described to you has been apparent since neurotransmitters were first recognized as being important. So people set out to have ways to measure neurotransmitters. And one of the first methods that was developed is a technique that's called microdialysis. And the idea here in microdialysis is you have a piece of microdialysis tubing or dialysis tubing. You flow liquid down the inside, and then you bring it back up, and through tubing, you collect it, and then you do some kind of analytical chemistry of that liquid, which has had the opportunity to interact with the brain extracellular fluid, and for the small molecules to diffuse across this membrane so you can get a measure of what's going on. This method has really been the workhorse of understanding what neurotransmitters do in behaving animals for the last 20 years. If you go to the literature and search on this, there are more than 10,000 papers that use microdialysis as their key word. So it's worth looking at some of the things that make it a good technique and a technique that people use. One thing is, once you've got substances out of the brain, you can analyze them by any method that you want to. Very popular is liquid chromatography with electrochemical detection, but you can use mass spectrometry, anything else. So you can get unequivocal identification of what you're looking at. The selectivity, therefore, is really excellent. One of the big problems of this technique is that the sampling's pretty slow. So if you flow through here, say at 1 microliter per minute, and the minimum volume that you can work with to do this kind of analysis is a microliter, you're going to have to sample on the minute time scale. And as you probably realize, things in the brain occur much faster than that. So you'd like a faster method. Detection limits are very low with this technique. And how do you know what detection limits you actually need with the method? Well, what you have to do is go to the literature and figure out what's the binding constant of a neurotransmitter with its receptor, because that's the level that is going to be the important concentration for actually doing the detection. Another limitation with this microdialysis is the probe size, and the dimensions, as you can see, are multiple micrometers. People have studied what happens when you implant these probes, and like someone was asking this morning, indeed they do cause damage in the rodent brain, and it's something to really worry about because you're sampling from damaged tissue. The electrochemical technique, which we use, actually one of the first things that we did when developing this was to make the probe as small as we could. And to do this, we use carbon fibers. That's supposed to be represented here. They're about 5 microns across. And when you go searching for the track left by this carbon fiber microelectrode, you have a very hard time finding it. Unless you do some sort of electrolytic lesion, the damage that's caused by this is pretty small. The carbon fiber is sealed inside a glass capillary, and that does actually have bigger dimensions, and it does cause more dimension, but that's far away for more damage, but it's far away from where you're actually making a measurement. So we're going to be sampling things using the electrochemistry that I explained to you a couple of slides ago. We've got pretty good selectivity. It's not as good as running a mass spectrometer. I'll show you how we achieve selectivity in a second. Our time scale is really good. It's on, say, the 10th of a second time scale in the measurements that I'll be showing you here today. So not maybe on the millisecond time scale you'd really like to be, but pretty good. The detection limits are in the nanomolar range, and that's just perfect for dopamine because that's the kind of concentration range where dopamine has interactions and activation of its receptors. And as I've said, the dimensions are quite small, not small enough so that we're in a synapse. We're certainly sampling in the extracellular fluid, but it turns out that dopamine isn't restricted to synaptic communication. It does communication what's called volume neurotransmission. That is, it interacts with receptors, transporters, so on, outside the synapse. So, these are the sorts of things you have to think about when you're planning to use a particular approach for measuring neurotransmitters. How often do I need to sample? Will millisecond resolution, is that what I need? That's what the time scale at which neurons are firing. I think you'll see that we do pretty good with a 100 millisecond time scale. The size of the probe certainly has to be small enough to be not causing a lot of damage, but maybe doesn't necessarily have to be inside the synapse to get really interesting information. What is critical is getting good chemical selectivity. That for many years was a weakness of this electrochemistry technique that I'm going to tell you about. We're good today, but the early experiments were really weak, and people were confusing whether they were looking at dopamine or actually ascorbic acid. Turns out there's a ton of ascorbic acid in the brain, and this is a molecule that can be easily oxidized. So if you read some of the 70s and 80s literature, there were violent debates raging back and forth. Is it ascorbate? Is it dopamine? What I'm going to show you today certainly is dopamine. And then sensitivity. Electrochemistry is not really a very sensitive technique, and I'll show you one of the ways in which we've achieved an additional degree of sensitivity that puts us down in the range that you need physiologically. Okay, so the way we get the selectivity we need is to use something called cyclic voltammetry. Take this carbon fiber electrode, and we apply a voltage to it in the form of a triangular wave. This wave takes about 10 milliseconds to execute. We go to more oxidizing potentials and then bring it back, and we repeat it every 100 milliseconds. When we scan positive, we take dopamine and oxidize it. Once we scan back negative again, this orthoquinone which we form can be reduced back to dopamine again. So you look at the current while you're measuring the voltage in this cyclic voltammetry technique, and if you did do that and just looked at the current while you were scanning the voltage, what you would see is a large amount of current that actually arises from charging the double layer of the electrode. And if you did the scan again while you were in a solution of dopamine, you might see a little bit of change in signal right here, but it's very unimpressive. So in fact, what you have to do is have a situation where you know there is little dopamine around, another situation where you know there is some dopamine around, and then you do a subtraction, and then you get this cyclic voltammogram that you see right here. So this is the oxidation of dopamine, and this is the reduction of dopamine. The amplitude here is proportional to the concentration of dopamine. Where those peaks are and their separation is a thing that allows you to identify this as dopamine. We've surveyed multiple molecules in the brain. The only compound in the brain that we found that has a cyclic voltammogram that looks like this is norepinephrine. Norepinephrine's a molecule which has a very similar structure. But to distinguish norepinephrine from dopamine, we take advantage of the fact that certain regions of the brain use dopamine as a neurotransmitter, other regions use norepinephrine, and so by our anatomical placement, we use that as well to help us in knowing exactly what neurotransmitter we're looking at. Okay, so the way a typical experiment would go, this might be an anesthetized animal or in a freely moving animal. What we do is we take a stimulating electrode and we implant it down into the cell body region where dopamine neurons are. This was to be a freely moving animal, on the day the animal's anesthetized, we would glue this electrode in place. Then we would take our carbon fiber electrode and put it down into a region where dopamine nerve terminals are. And the experiments I'll show you, there's in the nucleus accumbens. For freely moving animals, we actually implant a little hub up here on the animal's skull, and this hub is used to attach a micromanipulator, and the micromanipulator is used to lower a fresh electrode each day so we have a nice clean surface for doing these kinds of measurements. So the idea here is if you stimulate dopamine neurons, we know that they support action potentials and so forth, we'd expect that stimulation to propagate down the line and to cause dopamine release. So what we would really like to do is measure spontaneous dopamine release. In fact, we can do that, and I'll show you that in a minute. But we always start with stimulated release to make sure that we have our electrode positioned in a place that supports robust dopamine release. So here's going to be a typical stimulation experiment, okay? And this, again, is a set of 150 cyclic voltammograms. You can see it's recorded over 15 seconds. This axis is the voltage, so that's the apex of the triangle in the cyclic voltammetry waveform. This axis is acquisition time, and now I'm showing you the current in false color. So what you're actually doing is looking at 150 cyclic voltammograms simultaneously, so you can see what's going on through this whole experiment. Right here at this red bar, we stimulated dopamine neurons for about half a second, okay? So you can see before there was any stimulation, the signal's very quiet and very, nothing much is going on. When we do the stimulation, you see a peak emerge here and here, and then at later times you see other things emerge in the signal. So you can analyze what happened as a function of time by just simply taking a cut through here. And if you do that, we put the cut here because that's the potential where dopamine is oxidized. You can see when the stimulation is turned on, the signal goes up, and when it's turned off, the signal goes back down again. If you take the potential and make a cut across here and look at the current, this is something that's clearly not dopamine because we're at a very low potential here. This something goes along, and when you do the stimulation, it doesn't really change, but it changes rather after the stimulation and shows a big decrease in current. So to figure out what these substances are that we're looking at here, you can take cuts in the opposite direction, okay? So if we take a cut right here during the stimulation, you see now a cyclic voltammogram that has the peaks in the place where dopamine is supposed to be. So what we're looking at back here, that's dopamine changes. If you look at the cut out here at later times, you get a cyclic voltammogram that looks nothing like dopamine. It turns out that this particular cyclic voltammogram arises from a change in pH. So, and it actually represents a basic pH change. Now, hydrogen ion isn't actually reduced or oxidized at the potentials we're looking at. But you remember that big background that I showed you? The pH change causes the background to shift a little bit, and we get a pH signal here. This is why I put in the title Metabolites, because this pH change that you're seeing here arises from a metabolic change that's counterbalanced by changes in blood flow. And this turns out to be a really interesting signal, as we've heard today. These blood flow changes are the basis of MRI. So this, what you might have at first thought of as an interference, is actually a very interesting signal because it's telling you not about neurotransmission, but rather about metabolism and ongoing processes that are occurring there. So I'm going to focus on the dopamine today, but that signal you'll keep seeing cropping up in the examples that I show you. Okay, so once you have the information that this arises from pH, you can correct this signal for any pH contribution, and you get this signal that you see here. And we've been able to show that the rise that you see here, that's the rate of release of dopamine caused by the particular frequency of stimulation that we used in this example. You can take this part of the curve, actually fits perfectly to Michaelis-Menten kinetics because that process is governed by uptake by the dopamine transporter. And you may know that's a very interesting molecule. It's a target for many drugs of abuse like cocaine and amphetamine. So looking at how this changes with those drugs is certainly an interesting thing. Okay, so in the last year, we developed a program to put all this stuff into one package. It's available for free at our website. And so while you're an investigator doing the experiment, you can look at these things like how the current's changing at a particular potential, how the color plot is changing, what the cyclic voltammetry is doing. You control the stimulus frequency and all that kind of stuff. One of the really nice things about this program, and the reason I'm telling you about it, is the way I just talked you through the analysis of the data and how we could identify dopamine, sort of a crude approach. A much better way of doing things is to use the technique of principal component regression, whereby you generate a calibration curve using various different concentrations of dopamine. And in the case of the pH changes, various different pH changes measured with our device. Make sure that this has the shape that you expect, and then you can take this principal components and apply them to the signal that you measured in vivo and separate things out into a dopamine component, into noise components in the form of a residual, and so on. And so all of the data that we use today uses that particular approach. And so here's an example of how you could really clean up data. This was actually data obtained in a behaving animal, and a stimulation, a very mild stimulation was done right here, and you can see the dopamine change. But you can see a massive pH change over here. When you do the principal component analysis on it, you can see the stimulated dopamine release. You can see spontaneous dopamine release events. You can see the pH changes, and I find it really cool that all the noise ends up over here in the residual. So it's a way of sort of cleaning up some of the noise and getting some of the noise out of it. So having this kind of analysis was really important to us for separating out these overlapping signals. All right, how about sensitivity? How do we get the necessary sensitivity? Well, empirically, we discovered that if we took the potential limits of that triangular voltage scan, before we were scanning to a limit of about 1 volt, if we increased that to a more positive value, for example, 1.4 volts, the first thing we noticed was the background got a lot bigger. So that doesn't sound very good. But if you look at the signal, the background subtracted signal for dopamine when you do this, you see you get a dramatic increase in the signal for dopamine. So it's exactly the same concentration of dopamine in these two cyclic voltammograms. There's just we're scanning to a more positive voltage in the larger one. So these are the calibration curves for dopamine with the two different waveforms, here where we scan to 1 volt, here where we scan to 1.4 volts. The reason you get more signal to scanning to more positive voltages is you promote the adsorption of dopamine to the electrode. So this is the dopamine coming up to the electrode and sort of sitting and pre-concentrating on the surface of the electrode. We worried that this might distort the time scale of changes, and you can see they don't come all the way back to 0, but you can see that the general shape, and this was done in a solution environment, is about the same. So you don't lose that much by scanning to more positive potentials and having an adsorption-based signal. We haven't proved what's going on here, but this is a guess of what's going on. When you scan out here to say 1 volt, like this, you don't see much current here, but when you scan to 1.4 volts, you start to see current in the background. And we think that this current probably corresponds to the oxidation of the surface. The oxidation of the surface would lead to kicking off carbon dioxide from the surface and lead to functional groups being restored to the surface that could serve as the sites for this dopamine adsorption. So that's a speculative mechanism. It certainly works, though, for getting the concentration that we need to be able to look at the nanomolar concentration limits that are functional and work for dopamine. Okay, so the trick that electrochemists do is if they want to look at different things, they will change the waveform. So in our lab, we were interested in oxygen. I told you that pH signal comes from metabolism and changes in blood flow. Well, oxygen changes would have those same changes. And it turns out you can detect oxygen by scanning to negative potentials. So that's what the waveform is that is used here. We've scanned to 0.8 volts to detect catecholamines. We've scanned back to minus 1.4 volts, a potential that's sufficient to reduce oxygen, and then back to 0 again. And the overall cyclic voltammogram, when we do that, if we had a solution of catecholamine, in this case it's norepinephrine, there's the norepinephrine oxidation, reduction of its orthoquinone, and here is the reduction of oxygen. So if we do this just in a flow injection analysis system and put in a square wave of norepinephrine, here's a square wave of norepinephrine that we would get by analyzing the current change right here. The oxygen comes out upside down because the current is a reduction for looking at oxygen, negative current, where it's oxidation or positive current for looking at norepinephrine. So we can flip this current, calibrate this oxygen signal with known concentrations of oxygen, and now we have a way of simultaneously looking at norepinephrine and oxygen. And I'll come back to this in a little bit because norepinephrine, at least in the periphery, is a potent vasodilator. We'd like to see in the central nervous system what it does to oxygen levels. So this is a movie. This, if you were in my lab doing these experiments, you'd get to watch the color plots go by in real time. And in this particular movie, and this is an anesthetized animal, we're using that oxygen waveform. So oxygen's detected right here. So at this time, the dopamine neurons were stimulated. There's the dopamine signal, those two peaks. Here's the oxygen signal, and you can see the biggest oxygen signal occurs at a delayed time, just like that pH signal that I showed you, and that's because they're linked to the same thing. Here's another spontaneous oxygen change. So having these real-time view of what's going on around the tip of your electrode through these color plots allows the people in my lab to really position the electrodes in the places where there's something interesting to look at. When we're collecting this data, we might want to look at a single stimulation. That was what I showed you at first. If we want to if we want to do some signal averaging, we might want to look at stimulations that we did over and over again, and then we could signal average those together and improve the signal to noise, and so we can do that. But the really exciting part is in behaving animals, you don't ever know what's going to happen, right? So you'd like to have the collection just going on all the time. Our program will do that for us now. And what you're looking at down here are TTL signals, and these TTL signals come from a behavioral chamber. So we have our animal set up where he's pressing a lever or a tone is going off. And what we would like to see is what are the spontaneous changes that are going on during behavior in these kinds of situations. And we actually work with Regina Corelli, who's a psychologist at UNC, and has very elaborate behavioral routines that she's looking at with these rats. As was mentioned earlier today, dopamine is involved in reward-related processes. That's what Corelli would like to understand. So let me show you in a minute I will show you in a simple way how we can kind of get at dopamine's role in behavior. But anyway, so you generate this continuous record like this, and then our program lets you sort everything according to the TTL pulses, so you can generate a snippet. In other words, you do the same behavior over and over, and the snippet would be the average of each trial lined up either according to a cue or to a lever press or whatever it was that you're interested in. And this is easy to do with this. So I want to show you one behavior here and let you see how spontaneous dopamine changes occur. So this is, let me first show you the timeline of the experiment. So we've taught an animal, a rat, that we are going to play an audio-visual cue. In the movie that I'll show you, you'll see this as a light going on. There's also a tone, though. And that audio-visual cue lasts for 2 seconds. At the 2-second point, a lever pops out, and the tone and the cue continue. And the lever, if the animal goes and presses the lever, it delivers the electrical stimulation that I have shown you earlier to itself. Okay? This is an experiment known as intracranial self-stimulation. It's the same one that was talked about this morning with the dopamine study, except we have this extra part right here of this cue that predicts lever availability. So when the animal goes and presses the lever, some arbitrary time after the lever was available, you get the electrical stimulation, okay? So the electrical stimulation you already know causes dopamine release. I've shown you that. But what is of interest here is what happens when we present this cue. So this experiment, as I've tried to indicate, is repeated over and over again at random times so the animal can't predict when the cue is going to go on. So I'll just show you the result of a single trial here. You can see the rat here. The rat is just sitting there basically doing nothing. You can see there that is actually the manipulator that we use to lower the electrode into the brain. The electrode's now down in the ventral striatum, the nucleus accumbens, in a place where we know there's dopamine around. And so we want to see what's going to happen to dopamine during this experiment. So the blue trace down here is the dopamine, and there is when the cue came on, and there is where the electrical stimulation. So you understand this dopamine right here, which came on, is spontaneous dopamine. It has nothing to do with the stimulation. Rather, it's a learned signal that the animal has that's serving as a cue to tell it, get ready, the lever's going to be pressed out. I'll let you see this again. No, I won't. Yes, I will if I get this right. Okay, so you see there's no light. There's the light. That's the cue. And then you saw the dopamine go up, and then he presses the lever, and there's the dopamine signal. So that's a single trial. You can see that there's clearly good signal to noise. We can average a bunch together and improve on that. So that kind of changes what everybody thinks about dopamine. Dopamine used to be the molecule, or so you could read in Time magazine, that made you feel good. And you can see from this, it may be that. I don't know. But what it also does is it is an anticipatory signal in your brain that says to you, hey, something good's going to happen in the future. Okay, so we've done lots of experiments like that. Those cue-evoked dopamine signals, they occur not only with this sort of artificial situation called intracranial self-stimulation, but with food reward, with drug reward like cocaine self-administration, various other things. So now, your curiosity says, oh, great, I can detect dopamine. I know what dopamine does. Now, what's it doing? Okay? That was a big question for us, and the classic way an electrophysiologist would address that would be to do single unit recording. So what you do in single unit recording is you have an electrode, you put it close to a cell, it then picks up the electrical activity of this cell, and you would make multiple measurements of this. If you look at it as a function of time, you might see a big neuron and some smaller signals here. Each of those signals is arising from an action potential arising from nearby cells. Well, the carbon fiber is perfectly suited to measure these action potentials. We just need to change the measurement mode. So to do cyclic voltammetry, remember I told you we're measuring current, so we have an I to E converter. All we need to do is have a switch here to our carbon fiber electrode that lets us then measure voltage. So in the time between scans, we can measure single unit activity, and in each of these triangular waves, we can measure the dopamine concentration from the cyclic voltammetry. So we have been doing that. It works very, very nicely. We do make the basic change that we now have 200 milliseconds between cyclic voltammetry scans. Our chemical resolution, temporal resolution is a little bit down, but we certainly get a good picture of the unit activity. We get the unit activity to be the same whether or not we're recording the cyclic voltammograms. Now, you need to think about this experiment a little bit so that you can understand what the information you get from it actually is. So this is a light micrograph of this ventral striatum we've been talking about. And we used immunohistochemistry to mark some things that you need to know about. So the green here is a stain for the dopamine transporter. So where there's green, that's where dopamine nerve terminals are. Purple was used to stain the medium spiny neurons, and so at this angle I can't see it, but I seem to remember there's some purple halos around there. Those halos are marking this cell body of a medium spiny neuron. So if I go to the literature and get a drawing of a medium spiny neuron and superimpose it on here, this is what a medium spiny neuron actually looks like, okay? So there's the cell body. We need to be next to that to be able to pick up the electrical activity. But these are all the dendrites where presumably dopamine is interacting, glutamate is interacting. But if I superimpose now on the same scale our carbon fiber electrode, remember the carbon fiber electrode is just going to detect what's released right next to its surface. The single unit activity is going to come about from neurons that are, say, within 50 microns or so of the electrode surface. So what we're getting is a very local view of what's going on. Okay, so with that in mind, we get these electrical signals out of here. We amplify and filter them. We look at their shape. This is done with commercial Plexon software for those of you who do this sort of thing. And you can see in this example, there are two shapes. So we sort them into these two different stacks. And then we plot them out on a timeline like this. So this is supposed to indicate a conditioned stimulus, say that tone that I showed you. This would be what cell A was doing with each one of those tick marks being a indication of a firing event. And then we could sum up all those firing events over repetitive trials to give us a histogram of the firing rate of what's going on. So we've done this in this intracranial self-stimulation experiment. We actually see 4 different patterns. So we see cells that increase their firing right after the cue comes on. And remember, we got a dopamine surge right when the cue comes on. And in this case, they sort of overlapped here, the one to the stimulation and the one to the cue. If you look around right before the lever press, you can see that there's an increase in firing rate in some neurons. The percentages are given, or the numbers are given up here. Some neurons show an inhibition around the time of the lever press, and some neurons show virtually no change in firing rate at all. And in fact, it's interesting, those that don't show a change in firing rate, there's no change in dopamine either. Okay, so you got these firing rate changes and you got dopamine changes, but what you've simply got is a correlation. So you need a way to be able to go a step further to say, is this dopamine release causing those firing rate changes? So to do that, we've devised the following approach. We have taken our electrode, and my laser pointer seems to be dying here. Let's try this one. We've taken our electrode and attached to it some barrels. And these barrels are about 1 micron in diameter at the tip, and we're going to use them to eject substances by the process of electrophoresis. And, iontophoresis. And so this iontophoresis technique, we can mount all this in a micromanipulator on the animal's head, and then we can put him back in there now with an assembly that lets us measure single unit activity, chemistry changes. The barrels contain antagonists, and these antagonists will let us block specific D1 or D2 receptors. This is just an example of what happens if we block one of the types of receptor, the D2 receptor, and what happens is the dopamine release actually goes up. We're going through all of our data now. We've got a large number that have been used with this technique, and the net result is going to be that we're going to be able to say what dopamine receptor was activated, how much dopamine it took to do it, and how much the firing rate changed for each of those changes. But that data is being worked up right now. It's on my computer, and I'm working it up when I have a little bit of time between talks. But I think hopefully that shows you the promise that this kind of approach can do it. So we have now what I like to call the three-way sensor, okay? Got the neurotransmitter, we got the firing rate of adjacent neurons, and we can find out the receptor type that's nearby by applying agonists and antagonists through those ionophoretic barrels. And rather than showing you what goes on in ICS, because we hadn't got that all worked out quite yet, let me show you another example. So this is supposed to be a neuron. Here's supposed to be a blood vessel. There are astroglia that are involved here. And so these changes in oxygen that I showed you earlier, they come from neurotransmitters interacting with the astroglia to cause changes in dilation of these blood vessels. So we wondered if we could use this ionophoretic technique to look at something I showed you earlier, norepinephrine and how norepinephrine may be affecting neurons. So these are some measurements in a different region of the brain, a region called the bed nucleus of the stria terminalis. Here's what the background looks like, absolute flat line where oxygen is. We can put into these barrels a marker, an electroactive marker, something that has no neurochemical effect, but which we can use to see when substances are ejected from the barrel. So this is the ejection of 4-methylcatechol, our electroactive marker, just let us know the barrel's working well, and there's no change at all in the oxygen signal. Here, we put out norepinephrine. Now, norepinephrine, we wondered whether it would cause vasodilation or vasoconstriction. Here it is, it caused both. Here there's an increase in signal, which would correspond to a decrease in oxygen. I'm measuring from this dashed line right here, followed by a decrease in signal, which would correspond to an increase in oxygen. So here you can see directly in the brain that norepinephrine, when applied in physiological amounts, here we used a concentration of 4 micromolar, and we know that because we could measure it directly with the electrode that's adjacent, will cause physiological changes. How do we interpret this? Well, we did the same experiment with norepinephrine injection into a brain slice. So brain slices continue to respire. They continue to exhibit metabolic properties. They differ from the intact brain in that they've been removed from the brain, so they don't have blood flow. So when we put norepinephrine into this tissue, absolutely no change in signal whatsoever, telling us that this arises from a change in blood flow. And of course, then we had to do a lot more to unravel this. This is now published in the Journal of Cerebral Blood Flow and Metabolism. Okay, so what I've tried to do here today is show you how we've taken this electrochemistry, made it into a sensor for neurotransmitters and metabolites, added single-unit electrophysiology, added what we call controlled iontophoresis, because we have these electroactive markers here, and it gives us a lot more capability of unraveling what neurotransmitters are doing during behavior. The future. Well, there's lots of things that can be done in the future. One of the things that I would really like to see done is to get this into an array format. We have at one position 3 pieces of information, but we would love to know what's happening at other locations. So we have been working with Greg McCarty, who's a bioengineer at NC State, making various different kinds of sensors that will allow us to take this and find out what's happened in multiple situations. But that's a direction, as I say, for the future. So let me just end here by showing you, giving the acknowledgements to the people who actually did the work. NIH paid for this. All the electronics were made. We have an electronics facility in the chemistry department. They made all of that stuff. So Beth Bucher here did the iontophoresis and looking at the oxygen changes with norepinephrine. Nina Oasen-White is doing the intracranial self-stimulation along with electrophysiology, along with the iontophoresis, and she's aided by this young woman who's an undergraduate just leaving the lab, and this is Nathan here, who's also involved in the ICS project. So thank you very much for your attention.
Thank you very much, Mark. So we're open for some questions here at this point. Yeah, good.
Yeah, so what you call the background is may be important too. The tonic dopamine is also, do you see how that being regulated over a longer time scale in terms of the overall?
You're exactly right. We're seeing half of the dopamine story. We see what happens when there's a surge or burst of firing of dopamine neurons. But that background signal is really hard to know what it is. And the sensor over on the far right, it says a tonic sensor, is something that we're working on right now. Sort of strip out all the dopamine with electrochemistry, let it relax and see how much flows in, and through a differential procedure try and get at the background stuff. The real excitement about dopamine in recent years has been what does it do during bursts, and that's what we're ready to answer. What it does tonically may come out of this. We have some information in those iontophoretic experiments I told you, because the firing rate of dopamine neurons changes when we put out a dopamine antagonist. It either goes down with D2 antagonist or goes up with D1 antagonist. So there's information there too. So we're sifting that out.
I was very impressed by the plot of the 2 peaks of dopamine, one anticipatory and the other through the lever press. How much time does it take for the learning to occur? Do you see the time evolution of that anticipatory?
That's a wonderful.
And the other question is, are there other why dopamine in that stage and not other neurotransmitters? Or is information about other neurotransmitters?
Yes, we haven't solidified anything for that.
Those are both wonderful questions. So the first one was, again,
how much time does it take
the learning time. So the reason in my lab, I'm an analytical chemist, we do intracranial self-stimulation is because the learning time is about an hour or 2. okay? And so when you start, you only get dopamine to the lever press. That's the electrically stimulated release. There is nothing at all to the cue. Over a period of time, that cue response, say about 2 hours, it grows in, and it looks like what I showed you. Now, if you do something like cocaine self-administration, you're talking about 12 days of training where you teach the animal to press a lever to get its own cocaine. Or if you're talking about acquisition of this kind of behavior with food pellets, again, it takes about a week or 10 days to train the animal. So those are the kind of experiments that are going on in the Corelli lab. Her students have more patience than mine and are psychologists and are willing to wait the time it takes to train the animals properly. Your second question was, well, how do you know it's just dopamine that's doing this? Well, I've shown you we can measure norepinephrine. I didn't show you in freely moving animals, but we can do that. So we did exactly the same experiment with norepinephrine neurons and asked the question, do you fire during intracranial self-stimulation? And you've never seen such stable, flat responses in your life. Nothing from norepinephrine until we did the following experiment. What we did was what's termed extinction. We let the animal press the lever, but it wasn't connected any longer to the electrical stimulation. The animal gets really angry, and starts hammering on the lever. What's wrong? What's broken? And then he gives up. During that time when he's hammering on the lever, saying what's wrong, the norepinephrine goes up. So it clearly shows that dopamine and norepinephrine are doing two distinctly different things during this ICS experience. And admittedly, we need to do every neurotransmitter in the brain, and we don't have a probe for all of them to fully answer your question. But
it's an excellent technique, very innovative. I'm wondering if there's any other metabolites and the neurotransmitters that, so like in neurodegeneration, the neurotransmitters talk to immune cells as well. They have receptors for those, especially in the brain, as well as other metabolites. I'm just wondering if there's any directions kind of focusing on neurodegeneration and the immune system as a contributing role.
We did work some years ago on a model for Parkinson's disease using 6-hydroxydopamine and looking at how dopamine neurotransmission changed as you had different extents of lesions of dopamine neurons. And what you find is that release goes down, but so too does the rate of uptake go down, because when you make lesions of dopamine neurons, you lose release sites and transporters. So the net effect is dopamine release stays pretty robust, even though you've lost 80% of your neurons, because the thing that's removing dopamine is gone as well as the release site. At about 80%, though, that balance no longer works properly, and then you really start seeing impaired dopamine levels.
No, really interesting talk. A couple of technology-related questions. The first is about the electrode. So thinking about the carbon fiber electrode, I guess the logical extension of that is a single carbon nanotube. Is there any reason you wouldn't want a smaller electrode and have less of your surface double layer? Second part of the question, or second question, is there anything physical besides anything besides just basic engineering and clean room issues that keep you from having 100 by 100 or 1,000 by 1,000 arrays of electrodes, and can your program handle that kind of data? How would you analyze it?
So we wrote the program, as I said, it was done last year, and thinking to the future, so we can do 24 electrodes, not several thousand, but so we're ready for that. So no, it just is we're not engineers, so we're not really good at putting all this together. I do see problems on adding those ionophoretic barrels to this, but I count on my engineering friends to figure out how to get that particular part done. Oh, nanotubes. There are papers in the literature looking at the electrochemistry on nanotubes. And I think that's really good, but they're usually composites of nanotubes on carbon surfaces. I think what you're talking about, and what I'd love to do, is have one nanotube, like the one, and I have not seen anybody do that yet. We haven't done that. I think that would be a real good thing. So you have to remember that electrochemistry signals are proportional to the area of the electrode. So you would lose some, but I still think you might be able to do it.
Would you like to speculate on the function of the pH change and why it lasts so long?
So yeah, we studied the pH change in relation to the oxygen change, and they both, they track each other. The oxygen change actually occurs a few milliseconds before the pH change, but the duration of them are very similar, and the direction in which they go. If you get an increase in oxygen, what you get is a basic pH shift. Basic means CO2 was carried out of the bloodstream and washed away. So the two track each other. So I think what we're seeing is just this balance between metabolism and blood flow, and that the things that control blood flow are slower than neuronal communication, and that's why it seems to occur at a later time than the actual neurotransmission event.
So do you think this reflects the fact that some astrocytes can actually detect pH changes and then signal to the blood vessels to relax, or
Certainly, and in the paper that I showed you on norepinephrine, we went through each one of the noradrenergic receptors and showed that each one, when deactivated, was playing a different role in norepinephrine, and norepinephrine is then signaling to astroglia through nitric oxide, and other messengers are causing what I'm showing. Yeah. So there's a long, complicated chemistry there, and we'd like to figure that out. One of my students, Lindsay here, the third one in on the front row, has just found on campus an MRI guy who wants to work with us on this. So I think it's going to be a lot of fun using our carbon sensor in an MRI machine, correlating what you get there.
Well, Mark, thanks again. This has been just terrific. And I'd like to ask Ralph Greenspan to come up and make a few closing remarks here at the end of what's been a very exciting day.
Well, I just want to thank everyone. Thank all of you who attended. Thank the speakers. Thank the other organizers, Terry Sanofsky, John Reynolds, and Mike Saylor, and the staff of the Qualcomm Institute. I think this has been a tantalizing day, because I think what this gives us is a glimpse and maybe even a vision for the future of how we actually may someday be able to get to do the goals of the B.R.A.I.N initiative. And I, for one, take great hope from all of this. I also invite you to keep an eye out for the next one of these that we will do, probably sometime next year. We're going to try to cover other areas that are germane to the B.R.A.I.N initiative, such as large-scale recording from various brains, neural prosthetics, medical and otherwise, and perhaps things along those lines as well, but we don't know exactly what at this point. In any event, once again, let me just say that I think this has been a great day, and I appreciate all of you who have attended. Thank you very much.